Stephanie Mendez.
I'm Christian Aguilar.
I'm Gilmar Mencia.
And I'm Phong Nguyen.
And our project is on Lagoon Metabolism, and our mentor is Heather McNair, and her faculty advisor is Dr. Mark Brzezinski, and we are in the marine biology department.
Yeah, so some basic information about our test site. As you can see, this is the UCSB Lagoon, and it's a naturally formed 37-acre large lagoon that houses a unique mixture of both saltwater and freshwater. But most of the saltwater just comes in from this area over here, which is kind of the dumping ground of the marine science building. But so uniformly, it's mostly freshwater, which kind of makes it an ideal place for us to conduct our studies. So we're studying dissolved oxygen, more specifically. And you might ask yourself, what's so important about dissolved oxygen in bodies of water? Well, in fact, dissolved oxygen kind of tells us really valuable information about the biological and chemical reactions that occur in the water. So an example of this is you can use methods of figuring out dissolved oxygen to identify the pollution in the environment. Or like in this example right here with the two ponds, it's actually the same pond. It's just one's been treated with a bacteria that's considered to be heterotrophic, so it kind of competes for the nutrients that the algae could possibly want and essentially starve it. But overall, it kind of just tells us the overall health of the water.
And our project goal is to identify whether the lagoon is heterotrophic or autotrophic by measuring the dissolved oxygen concentration in the lagoon. So right here is the scale from 0 to 10. And if the result we calculate is under 3, so we can say that the lagoon is heterotrophic. And anything above 3 ppm can be considered autotrophic environment. So you may wonder what are heterotrophs and autotrophs. Actually, autotrophs are organisms that can produce their output from substances available in their surrounding by using light, such as photosynthesis. And heterotrophs are totally opposite. They cannot synthesize their output and rely on other organisms, such as plant or sea animal for nutrition. Technically, the definition is autotrophs obtain the carbon from inorganic sources like carbon dioxide, while heterotrophs get the reduced carbon from other organisms. And if the process of reducing carbon is too slow, so there may be not enough oxygen in the water, and the environment turns bad for fish. And as the result of that, the fish may be dead or move away. So right here is the picture of heterotrophic environment. As you can see right here is the area, the green area along with the shore is considered heterotrophic. And the next picture is autotrophic environment where enough oxygen for fish to survive. The fact it is producing oxygen and reducing carbon dioxide.
So we took samples of water from the lagoon, and a few of the bottles were placed in sunlight, so they would go through photosynthesis and respiration. And the other bottles were covered in foil, so they only went through respiration. And we were trying to find the primary production levels of oxygen between the light bottles and the dark bottles. And in order for photosynthesis to occur, you need carbon dioxide, water, and light to produce sugar and oxygen. And respiration is the opposite equation.
Okay, well, the way we started our project is we went out to the UCSB Lagoon and we picked out 3 test sites. And as you can see from this picture right here, these are our test sites. So we got samples from the midpoint of the lagoon, the western side of the lagoon, and the eastern side of the lagoon. So once we collected our samples, we wrapped some of our samples with aluminum foil, as those were going to be our dark bottles. And right here, these were going to be our light bottles, and these were our time 0. So the only samples that we took back to our lab were the time 0s, and we began titration. And as you can see right here, our light bottles and our dark bottles were incubated for a period of 23 hours. So once we came back the next day, we picked up all of our samples and we began titration. So you might want to ask yourself, what is titration? Well, titration is a common method used in chemistry to determine the concentration of a base or an acid. So if you know the concentration of one thing, you can determine the concentration of another. So once we got our samples, we added manganese chloride and alkaline iodide, which made our sample into a brown precipitate. Now what that brown precipitate lets us know is that there's oxygen present in the water. And as you can see in the chemical reaction, that's where the oxygen is present right here. Afterwards, we added sulfuric acid to our sample, and that dissolved the brown precipitate into a more yellow precipitate. Now, what that tells us is that there was an immediate reaction between the potassium iodide that we added from the first reaction, and that liberated iodine. And at that point, we knew that the same ratio of iodine is the same ratio. It's a one-to-one ratio with the oxygen. So the same moles of iodine, it's equivalent to the same moles of oxygen at that point. So as you can see, the oxygen right here is being replaced with the iodine right here. So once we knew that, we began to titrate our sample with sodium disulfate. And since at this point, the oxygen was replaced, the only way that we can bring back the oxygen to a visibility point is by adding starch. And as you can see, it went from a light yellow into a more purple precipitate. So we further titrated the sample with sodium disulfate until it reached its endpoint, until a clear solution. At that point, from our depending how much sodium disulfate and reagents we added to our samples, we incorporated into our formula, which is right here. And this formula was incorporated into every sample. Once we had how much oxygen per milligrams per liter was in each sample, we got an average of those. For the light bottles, we have an average for the dark bottles, and we had an average for the time zeros. And that's how we got our net primary production, our respiration, and based on these numbers, we subtracted the net primary production minus the respiration to give us a gross primary production.
Okay, so using the numbers that we got from the previous slide and our concentrations, we can see that our time zero, our blue bar, we can use it as a reference point. So comparing it to the dark bottles, so the red and the green, we can kind of tell a lot about what's going on in the water. So our expectation was that since the darker bottles are only allowed to respirate and not really photosynthesize, that it would be lower than the reference point. And if you see here, even with an error bar of the standard deviation, so 68% of the time, it really never overlaps. So we can pretty firmly say that the water is respirating at a healthy rate, whereas the light bottle had a lot of overlap, and what that tells us is that either there was probably something going on in the bottle, or probably there's too much respiration going on in the water. So to add more detail to this, what we say is that through the calculation of our net primary production and our respiration, we can process this information through what's called the gross production respiration ratio. And essentially what that tells us is this right here, so percentage-wise, of how much oxygen is being used, in other words, the respiration, and how much oxygen is being produced or photosynthesized, in other words, the net primary production. And as you can see, there's more oxygen being used compared to what's being produced. So what that's kind of telling us is that the lagoon, on average, is heading more towards the heterotrophic side. And that's kind of strange because looking at it by observation, we see lots of fish and algae and such. So we kind of assumed it was going to be an autotrophic kind of from the start. So it's pretty interesting to see that our results kind of showed otherwise.
And future improvement. So with that being said, if we can collect more sample of the lagoon, so we can have better averages for our calculation. The next improvement is, since we already took sample of water from the edge of the lagoon, so in the future we just need to get more sample in the middle of the lagoon where larger plants are growing and producing more oxygen. And to work with the reaction faster, so we need to precap the bottle immediately after we put on the reagent so that less oxygen escapes. And we also have shorter incubation period, so that less oxygen will be lost due to overproduction of oxygen. If we have something like functional equipment, so that we can compare our calculation with that result oxygen reader to turn our calculation, to turn our result more accurate.
And we would like to thank our mentor, Heather McNair, for inviting us into her lab and allowing us to conduct research, and her faculty advisor, Dr. Mark Brzezinski, and also Dr. Anthony Carnes and Dr. Jens and all of the resident assistants and UCSB, the Cook Bridge program, and CSAC.
Thank you very much.